Hello students, today we will study about plant tissue culture. Plant tissue culture broadly refers to the in vitro cultivation of plants and various parts of the plant. That means organs like embryos, tissues, single cells, protoplast, etc. The cultivation process is invariably carried out in a nutrient culture medium under aseptic conditions. Plant cells have certain advantages over animal cells in culture system. Unlike animal cells, highly mature and differentiated plant cells retain the ability of totipotency, that is, the ability of change to meristematic state and differentiate into a whole plant. Now, let's know about the benefits of plant tissue culture. Plant tissue culture is one of the most rapidly growing areas of biotechnology because of its high potential to develop improved crops and ornamental crops. With the advanced with the advances made in the tissue culture technology, it is now possible to regenerate species of any plant in the laboratory. To achieve the target of creating a new plant or a plant with desired characteristic, tissue culture is often coupled with the recombinant DNA technology. The techniques of plant tissue culture have largely helped in the green revolution by improving the crop yield and quality. The knowledge obtained from plant tissue culture has contributed to our understanding of metabolism, growth, differentiation, and morphogenesis of the plant cells. Further developments in tissue culture have helped to produce several pathogen-free plants. Besides the synthesis of many biologically important compounds, including pharmaceuticals. Because of the wide range of applications, plant tissue culture attracts the attention of molecular biologists, plant breeders, and industries. Now, let's know about the basic structure and growth of a plant. An adult plant basically consists of a stem, root, and with many branches. Both the stem and root are characterized by the presence of apical growth regions, which are composed of meristematic cells. These cells are primary sources of all the cell types of a plant. The plant growth and the development consist of development occurs in two different ways. First is determinate growth and indeterminate growth. Let's know about determinate growth. This is characterized by cessation of growth as the plant parts attain certain size and shape. Example, leaves, flowers, and fruits. Now, second is indeterminate growth. What is indeterminate growth? This refers to the continuous growth of roots and stems under suitable conditions. It is possible due to the presence of meristems which can proliferate continuously. As the seed germinates and seedling emerges, the meristematic cells of the root apex multiply. Above the root apex, the cells grow in length without multiplication. Some of the elongated cells of the outer layer develop into root hairs to absorb water and nutrients from the soil. As the plant grows, root cells differentiate into phloem and xylem. Phloem is responsible for the absorption of nutrients while xylem absorbs water. The meristematic cells of the shoot apex divide leading to the growth of stem. Some of the stem cells differentiate and develop into leaf primordia and then leaves. Auxiliary buds present between the leaf primordia and elongated stem also possess meristems which can multiply and give rise to branches and flowers. Let's know about conventional plant breeding and plant tissue culture. Since the time immemorial, man has been closely involved in the improvement of plants to meet his basic needs. The conventional methods employed for the crop improvement are very tedious and long time process. Further, in the conventional breeding methods, it is not possible to introduce desired genes to generate new characters or products. With the development in the plant tissue culture, it is now possible to reduce the time for the creation of the new plants with desired characteristics, transfer of the new genes into plant cells, and large scale production of commercially important products. Now let's know about the terms used in tissue culture. First term is explant. What is explant? An excised piece of differentiated tissue or organ is regarded as an explant. The explant may be taken from any part of the plant body like leaf, stem, root. And second point is callus. The unorganized and undifferentiated mass of plant cells is referred to as callus. Generally, when plant cells are cultured in a suitable medium, they divide to form callus, that is, a mass of parenchymatous cells. Dedifferentiation, the phenomenon of mature cells reverting to meristematic state to produce callus, is differentiated. Differentiation is possible since non-dividing quiescent cells of explant, when grown in a suitable culture medium, revert to meristematic state. Now let's know about redifferentiation. The ability of the callus cells to differentiate into a plant organ or a whole plant is regarded as redifferentiation. Now, one next term is totipotency. What do you mean by totipotency? The ability of an individual cell to develop into a whole plant is referred to as cellular totipotency. The inherent characteristic feature of plant cells, namely dedifferentiation and redifferentiation, are responsible for the phenomenon of totipotency. Now let's know about the types of culture. These are different types of tissue culture techniques, mainly based on the explant used. First is callus culture. Callus culture involves the culture of differentiated tissue from explant, which dedifferentiates in vitro to form callus. Culture of isolated plant organs is referred to as organ culture. The organ used may be embryo, seed, root, endosperm, anther, ovary, ovule, meristem, or nucleus. The organ culture may be organized or unorganized. Let's know about organized organ culture. Organized organ culture means when a well organized structure of a plant, that means seed or embryo, is used in culture, it is referred to as organized culture. In this type of culture, the characteristic individual organic culture, organ structure is maintained, and the progeny formed is similar in structure as that of the original organ. Second is unorganized organ culture. This involves the isolation of cells or in tissues of a part of the organ and their culture in vitro. Unorganized culture results in the formation of callus. The callus can be dispersed into aggregates of cells and or single cells to give a suspension culture. Now let's know about cell culture. The culture of isolated individual cells obtained from an explant tissue or callus is regarded as cell culture. These cultures are carried out in dispenser medium and are referred to as cell suspension cultures. Let's know about protoplast culture. Plant protoplast, that is cells devoid of cell walls, are also used in the laboratory for culture. Now let's know about basic techniques of plant tissue culture. The requisite explants, that means buds, stems, seeds, are trimmed and then subjected to sterilization in a detergent solution. After washing in sterile distilled water, the explants are placed in a suitable culture medium, that may be liquid or semi-liquid, semi-solid form, and incubated. This results in the establishment of culture. The mother culture can be subdivided as frequently as needed to give daughter cultures. The most important aspect of in vitro culture technique is to carry out all the operations under aseptic conditions. Bacteria and fungi are the most common contaminants in plant tissue culture. They grow much faster in culture and often kill the plant tissue. Further, the contaminants also produce certain compounds which are toxic to the plant tissue. Therefore, it is absolutely essential that aseptic conditions are maintained throughout the tissue culture operations. Let's know about the brief about callus culture. Callus is the undifferentiated and unorganized mass of plant cells. It is basically a tumor tissue which usually forms a wound of differentiated tissues or organs. Callus cells are parenchymatous in nature, although not truly homogeneous. On careful examination, callus is found to contain some quantity of differentiated tissue besides the bulk of non-differentiated tissue. Callus formation in vivo is frequently observed as a result of wounds as the cut edge of stems or roots. Invasion of microorganisms or damage by insect feeding usually occurs through callus. Now let's know about explant of callus culture. The starting material, that is explant for callus culture, may be the differentiated tissue from any part of the plant, that may be root, stem, leaf, anther, flower. The selected explant tissues may be at different stages of cell division, cell proliferation, and organization into different distinct specialized structures. If the explant used possesses meristematic cells, then the cell division and multiplication will be rapid. Now let's know about factors affecting callus culture. Many factors are known to influence callus formation in vitro culture. These include the source of the explant and its genotype, composition of medium, MS medium most commonly used, physical factors, temperature, light, etc., and growth factors. Other important factors affecting callus culture are age of the plant, location of explant, physiology, and growth conditions of the plant. Now let's know about physical factors. What are the physical factors? The temperature in the range of 22 to 28 degrees centigrade is suitable for the adequate callus formation. As regards the effect of light on callus, it is largely dependent on the plant species. Light may be essential for some plants while darkness is required by others. Growth regulators. The growth regulators to the medium strongly influence callus formation. Based on the nature of the explant and its genotype and the endogenous content of the hormone, the requirement of growth regulators may be categorized into three groups. First is auxin alone, cytokinin alone, both auxin and cytokinin. Let's know about suspension culture from callus. Suspension culture can be initiated by transferring friable callus to liquid nutrient medium. As the medium is liquid in nature, the pieces of callus remain submerged. This creates an aerobic condition and ultimately the cells may die. For this reason, suspension cultures have to be agitated by a rotary shaker. Due to agitation, the cells gets dispersed besides their exposure to aeration. Now let's know about applications of callus cultures. Callus cultures are slow growth plant culture systems in static medium. This enables to conduct several studies related to many aspects of plants growth, differentiation, and metabolism. First point is nutritional requirement of plants. Second is cell and organ differentiation. Third is development of suspension and protoplast cultures. Fourth is homoclonal variations. Fifth is genetic transformations. Production of production of secondary metabolites and their regulations. Let's know about cell culture. The first attempt to culture single cells was made in as early as 1902 by Howard Land. Although he was unsuccessful to achieve cell division in vitro, his work gave a stimulus to several researchers in later years. Good success was achieved not only for cell division but also to raise complete plants from single cell cultures. Let's know about applications of cell cultures. Cultured cells have a wide range of applications in biology. Elucidation of the pathways of cellular metabolism. Second point is serve as good targets for mutation and selection of desirable mutants. Third is production of secondary metabolites of commercial interest. Fourth is good potential for crop improvement. Now let's know about cell culture techniques. The in vitro cell culture technique broadly involves some aspects. Let's know about these aspects. First is isolation of single cells. Second is suspension cultures, suspension culture, growth and subculturing. Third is types of suspension cultures. Fourth is synchronization of suspension cultures, measurement of growth of cultures, measurement of viability of cultures cells. Now let's know about isolation of single cells. The cells employed for in vitro culture may be obtained from plant organs and from cultured cells, cultured tissues. From plant organs, plant leaves with homogeneous population of cells are the ideal sources for cell culture. Single cells can be isolated from leaves by mechanical or enzymatic methods. Let's know about mechanical method. Surface sterilized leaves are cut into small pieces that is less than 1 centimeter. Less than 1 centimeter square, suspended in the medium and subjected to grinding in a glass homogenizer tube. The homogenate is filtered through filters and then centrifuged at a low speed to remove the cellular debris. The supernatant is removed and diluted to achieve the required cell density. Enzymatic method. Let's know about the enzymatic method. The enzyme macerozyme can release the individual cells from the leaf tissue. Macerozyme degrades middle lamella and cell wall of parenchymatous tissues. From cultured tissues. From cultured tissue, single cells can be isolated from callus cultures, that is, growth from cut pieces of surface sterilization, plant parts. Repeated subculturing of callus on agar medium improves the friability of callus so that the fine cell suspensions are obtained. Suspension cultures. Let's know about suspension cultures, growth and subculture. The isolated cells are grown in suspension cultures. Cell suspensions are maintained by routine subculturing in a fresh medium. For this purpose, the cells are picked up in the early stationary phase and transferred. Here 3. As the cells are incubated in suspension cultures, the cells divide and enlarge. The incubation period is dependent on 3 broad categories. First is initial cell density, second is duration of lag phase, third is growth rate of cells. Now let's know about types of suspension cultures. There is two types of suspension culture. One is batch culture and second is continuous cultures. Let's know about batch culture. The batch culture is a cell suspension culture grown in a fixed volume of nutrient culture medium. In batch culture, the cell division and cell growth coupled with an increase in biomass culture occur until one of the factors in the culture environment becomes limiting. The cells exhibit the following 5 phases of growth when the cell number is suspension culture is plotted against the time of incubation. Let's know about lag phase. Lag phase, first point is lag phase, that is characterized by preparation of cells to divide. Second is log phase, that is exponential phase, where the rate of cell multiplication is highest. Third is linear phase, that is represented by slowness in cell division and increase in cell size expansion. Fourth is deceleration phase, characterized by decrease in cell division and cell expansion. Fifth is stationary phase, represented by a constant number of cells and their size. Let's know about continuous culture. In continuous culture, there is a regular addition of fresh nutrient medium and draining out the used medium so that the culture volume is normally constant. These cultures are carried out by the specially designed culture vessels. Continuous cultures are carried out under defined and controlled conditions. Cell density, nutrients, oxygen, pH, etc. The cells in these cultures are mostly at an exponential phase, that is log phase of growth. Continuous cultures are of two types, that is open and closed type. Let's know about open continuous culture. In this culture, the inflow of fresh medium is balanced with the outflow of the volume of suspended medium. Along with the cells, the addition of fresh medium and culture harvest are so adjusted that the cultures are maintained indefinitely at a constant growth rate. At a steady state, the rate of cells removed from the cultures equal to the rates of formation of new cells. Now let's know about open continuous culture. Open continuous culture system is regarded as chemostat if the cellular growth rate and density are kept constant by limiting the nutrient in the medium, that is glucose, nitrogen, phosphorus. In chemostat cultures, except the limiting nutrient, all the other nutrients are kept at high concentrations. As a result, any increase or decrease in the limiting nutrient will correspondingly increase or decrease the growth rate of cells. Let's know about closed continuous cultures. In this culture, the cells are retained while the inflow of fresh medium is balanced with the outflow of corresponding spent medium. The cells present in the outflowing medium are separated and added back to the culture medium. As a result, there is a continuous increase in the biomass in closed continuous cultures. These cultures are useful for studies related to cytodifferentiation and for the production of certain secondary metabolites, example polysaccharides and coumarins. Thank you. Any questions regarding this? You can ask me in the comment box. I'll clear all the questions and all the doubts I'll clear. Please subscribe my channel. Thank you.